Maybe, yeah, I could do that actually. Right, right. Okay, thank you. Okay, so far I've talked to you a little bit about, basic chemistry review, organic chemistry review, and talked a little bit about molecular visualization software, and I've introduced you to a couple of tools, PyMol and Chimera, for visualizing molecular structures in 3D and exporting reference images that you could then illustrate in Illustrator. So basically, at this point, you know how to illustrate individual molecules, at least in Illustrator. In a couple of weeks, we'll come back and we'll talk a lot more about illustrating them in 3D, actually rendering them directly in Chimera, but more importantly, exporting them in a 3D format that you can then bring into something like Maya or Cinema 4D. I'll introduce you to just a little bit of Cinema 4D for doing some more sophisticated rendering of molecular structures. But so far, all you've illustrated is just a static molecule, just kind of sitting there, not doing anything. And occasionally you will have clients who are going to ask you to do that, to just create an illustration of a molecule. But it's much more likely that a client is going to want you to illustrate the role that that molecule plays in some kind of important cellular process, some kind of molecular reaction, molecular pathway. So I want to talk a little bit about illustrating some of these cellular and molecular processes. Now, there's no way that I can cover the whole range of different kinds of molecular interactions that take place inside a cell or inside a body. You'd have to take a whole course in biochemistry and another whole course in molecular biology to get that level of detail. So I can't possibly cover everything, but I did want to introduce you to certain specific cellular processes that I've been asked to illustrate a lot. And I think these are some of the things that you're most commonly going to be asked to illustrate. And specifically, I want to talk about these concepts here, the transport of certain molecules, especially ions, but not just ions, the transport of certain molecules in and out of the cell. And in some cases, that's passive transport that doesn't require any energy. In other cases, it's active transport that actually requires energy, usually in the form of ATP, to drive the movement of molecules across the cell membrane, either into or out of the cell. I want to spend some time talking about different kinds of receptors. So how is it that a small molecule binds to a protein on the surface of a cell and then triggers some kind of cellular reaction? This is a very common thing that you might be asked to illustrate. And there's a whole bunch of different categories of receptors that I'll talk to you about. Related to that are what are called second messengers, which is another aspect of cellular signaling. So oftentimes what happens is you'll stimulate a receptor on the surface of the cell, and then that receptor in turn triggers these second messengers that then amplify the reaction and carry that reaction into the cell to create some kind of response in the cell. Oftentimes that would be gene transcription, where the second messengers turn on the transcription of specific genes in the cell. We'll talk a little bit about activation of receptors versus inhibition of receptors. And then at the very end, I'll talk about how to actually illustrate these things, just different depictions of receptors, different ways to illustrate them. as well as depictions of cell membranes, because many of these receptors, not all of them, but many of these receptors are things that are actually embedded in the cell membrane. So I'll talk a little bit about different ways of illustrating the membrane. So starting with this list, starting with our outline here, just want to talk briefly about passive transport. So in passive transport, molecules are flowing along their concentration gradient. In other words, molecules are flowing from an area of high concentration to an area of low concentration. That kind of movement doesn't require any energy. Molecules will naturally move from an area of high concentration to an area of low concentration. However, the cell membrane represents an impermeable barrier to many kinds of molecules, especially any kind of molecule that has a charge, like an ion, any kind of molecule that's polar, like water, can't cross the cell membrane because the lipid tails, the lipid part of that cell membrane is hydrophobic, and it doesn't allow anything charged or anything polar to cross the membrane. So oftentimes, in order to allow this passive transport to take place, you need some kind of opening in the membrane, which would be something like a pore or some kind of channel that simply allows molecules to pass easily from the outside to the inside, or the other way around, to pass from the cytoplasm inside the cell to the outside. So here's one example. This is a protein called aquaporin. And on the left, you're looking at what I've called a top view, in quotes, meaning the extracellular view. It's as if you're outside the cell looking down on this protein as it's embedded in the cell membrane. And then on the right-hand side, you see essentially a side view, a cutaway view of the aquaporin molecule. And that gray bar in the background represents the cell membrane. So this protein molecule is embedded in the cell membrane. And I think you can tell in cross-sectional view that there are a couple of channels passing through the molecule. You can see them on the left side too, as those little holes, those little 4 little round holes that allow water to pass in or out of the cell, depending on whether the cell is, depending on the osmolarity, whether it's hypotonic or hypertonic. And then water simply flows passively without needing any energy. Here's another example, the glucose transporter. It's another protein that's embedded within the cell membrane. And that space filling model that you see up there, with the red and gray and white atoms, that's a molecule of glucose. And that molecule binds to the outside of the protein. And then there's actually a shape change in the protein that then allows the glucose molecules to pass to the inside. There are actually, if I remember correctly, there are 2 different binding sites. One glucose is bound to the outside, one is bound to the inside. When that second glucose binds to the outside, then the shape change happens and the one on the inside is released. This actually does not require any energy because the glucose is moving along its concentration gradient from high to low. And the reason that the concentration of glucose is low on the inside of the cell is because the cell is continuously using glucose for energy. So that glucose is constantly being broken down by glycolysis and by the Krebs cycle to generate energy for the cell. So you're always depleting the glucose on the inside. So that means that the concentration is almost always higher on the outside of the cell. So the glucose can just flow passively through this transporter molecule to the inside. Now, in addition to passive transport, where molecules flow with the concentration gradient, where they're flowing from high to low, there are also proteins embedded in the cell membrane that can actively pump ions or molecules the other way against their concentration gradient. So they're actually pumping molecules from an area of low concentration towards an area of high concentration, which would not happen spontaneously. And it actually requires energy to pump something against its concentration gradient. So that energy is usually in the form of ATP. So these ion pumps require ATP to pump ions a certain direction. This image here is the calcium pump, or a particular calcium pump. There are several of them, but this is one particular calcium pump that's found in muscle cells. And once again, the gray box represents the cell membrane. So you can see which part of this molecule is actually embedded in the cell membrane. And you see that then there's a small intracellular part at the bottom that extends a little bit into the cytoplasm. And then there's a much larger extracellular component that sits outside the cell, outside the cytoplasm. Now, this particular calcium pump is important in muscle contraction. I don't know if you recall from an A&P class, from physiology class, but calcium is an important ion that triggers muscle contraction. And I talked about this in our previous class. I was showing an example of a molecule of actin, myosin, and tropomyosin. So I described the process a little bit then. So you have calcium ions that are released into the sarcoplasm, which is just a fancy name for the cytoplasm of a muscle cell. So calcium ions are released into that sarcoplasm, and the calcium ions bind to a protein called tropomyosin, cause a shape change in the tropomyosin, which moves the tropomyosin a little bit, and that exposes binding sites on the actin thin filament. Then the myosin heads from the myosin thick filament bind to those binding sites on actin, and then the myosin heads will then contract to generate that contractile force, that pulling force of muscle. Now, if calcium ions simply flooded into the cell and bound to the tropomyosin, if those calcium ions just stayed there in the sarcoplasm, the muscle would remain permanently contracted because the myosin heads would be bound to the actin, and they'd never let go. And your muscle would just tighten up and cramp up. And actually, that's one reason people get cramps, is because if you don't have adequate blood flow and circulation to a muscle, you don't have any way of getting rid of that excess calcium in the cell. So what this molecule does is this molecule is responsible for pumping the calcium ions back out of the sarcoplasm, out of the cytoplasm, back into something called the sarcoplasmic reticulum, which is kind of like the endoplasmic reticulum of a cell. So it moves the calcium ions out of the cytoplasm into the sarcoplasmic reticulum, where those calcium ions are then stored. And the next time you have a muscle contraction, then the calcium ions are released again. But so this is what then is responsible for the relaxation of a muscle after it contracts. Some other examples of proteins that are involved in active transport, moving molecules against their concentration gradient, are things called symporters and antiporters. Just going back to the ion pump, in the case of the ion pump, this protein is only moving one thing. It's moving just calcium ions. In the case of symporters and antiporters, the molecule is moving two things at once. In the case of a symporter, the molecule actually has to move two molecules together simultaneously through the protein to the other side. So a symporter, sim meaning like symbiosis, meaning two things together. Two molecules have to move together through this protein. And then there's an example of that in the lower left. There's the lactose H+ symporter. So that's going to bring lactose, which is a sugar found in milk. For those people who are lactose intolerant, you're probably familiar with that. And it also moves a hydrogen ion, H+, has to move these things together simultaneously. And again, it uses ATP to generate the energy to move both the lactose and the hydrogen against their concentration gradient from the outside of the cell into the cell. Something similar is what's called an antiporter. In an antiporter, the molecule is moving two things, but it's moving one molecule one direction while simultaneously moving a different molecule the other direction. And there's an example of that here called the glycerol phosphate antiporter. So it's moving a molecule of glycerol. Actually, it looks from the picture, it looks like glycerol phosphate moving one direction, whereas then there's a phosphate group moving the opposite direction. And then probably the most famous, the most well-known one that you may be familiar with if you reach back and think back to your anatomy and physiology class, or just a general physiology class, is the sodium potassium ion antiporter, where I forget the exact numbers. What is it? 2 sodiums move in for 3 potassiums moving out, or something like that. There's 2 of one going one way and 3 of another going the other way. But this is an important protein for maintaining the polarity of the cell membrane in a neuron, where the outside of the cell membrane of a neuron is a little more positive and the inside is a little bit more negative. So it must be moving 3 positive charges out and 2 positive charges in to end up with a net positive charge on the outside. Okay, so these are, you've got pores that just allow for passive transport. You've got pumps, symporters, and antiporters that use energy to move molecules against their concentration gradient. Then we move into the category of molecules called receptors. And there are lots of these and lots of different kinds. So I'm going to walk you through some of the most common kinds. I'm probably not going to cover them all. You know, there's probably a few that I'm going to miss, but these are ones that I've been asked to illustrate many times, and ones that I think you're going to run across quite often in your career, assuming you do any of this kind of cellular illustration, and most likely you will. So one category of receptors are what are called ligand-gated ion channels. So let's dissect that word. Let's look at the last part of it first. It's an ion channel, meaning it's a molecule that allows ions to pass across the cell membrane. So charged atoms like sodium plus, potassium plus, calcium 2 plus, and so on. So it's some kind of molecule that's allowing ions to pass through the cell membrane. But the first part of the word, the first part of the name, ligand-gated. So a ligand is a molecule that binds to a receptor. They're often small molecules, but not always. There are some proteins that function as ligands, but the ones that you may be most familiar with are generally small molecules. This would include the neurotransmitters like serotonin or acetylcholine or norepinephrine, dopamine, GABA, glutamate, all these different neurotransmitters. These are all examples of ligands. So these are all small molecules that bind to a receptor to then cause the receptor to do something. So a ligand-gated ion channel means an ion channel that opens up. That's where the word gated comes in. There's essentially a gate inside. The gate opens in response to the binding of a ligand. So here's a good example of that. This is one example of an acetylcholine receptor. There are different kinds of acetylcholine receptors. This particular one is known as a nicotinic acetylcholine receptor. So acetylcholine binds to this receptor, and you can actually see, I believe that this acetylcholine shown in red, binding to its binding pocket on the receptor. And in response to the binding of acetylcholine, there's a subtle shape change in the interior of the molecule that now opens up a channel in the middle of the molecule to allow sodium ions to flow through. And the sodium ions will flood from the outside of the cell. They'll come rushing into the inside of the cell. If you look at the structure of it, if you look at the picture on the right, you see that there are 5 subunits. Trying to remember, what is it? 2 alphas, 2 betas, and a gamma, I think. So there are 2 that are sort of turquoise in color, 2 that are kind of orange in color, and then there's one that's like a darker blue. So a total of 5 subunits arranged in this kind of star shape, or what we call radial symmetry. And then if you look at the side view to the left of that, again, the gray box represents where the cell membrane is located. So this thing's embedded in the cell membrane. So the acetylcholine binds to the outside of the receptor, to the extracellular part, causing a shape change in the protein, opening up that pore in the middle to allow sodium ions to flood through. This is a very common receptor that's found in lots of synapses. So lots of synapses between two neurons. The presynaptic neuron releases acetylcholine into the synaptic cleft. The acetylcholine then binds to this receptor on the postsynaptic neuron. That causes this flood of sodium ions to rush into the cell, and that can initiate an action potential in the postsynaptic neuron. So then that leads to the propagation of a nerve impulse in the postsynaptic neuron. It's also the receptor that you find at the neuromuscular junction. So same kind of thing, you have a nerve that is innervating a particular muscle. And by what we mean by that is that when there's an action potential that comes down through that nerve then releases acetylcholine into the neuromuscular junction. The acetylcholine diffuses across that gap, binds to this molecule. The channel opens and sodium ions rush into the muscle cell this time, and that creates an action potential in the muscle cell, which eventually then opens the calcium channels that allow muscle contraction to take place. Here's another view of that. By the way, most of these are David Goodsell's illustrations. I've talked about him a handful of times, so you start to recognize his distinctive style. Yeah, Sheila.
There's ways to sort of because his style is so replicated, right? Like, you kind of see this.
People try. I mean, I've tried. I'm not really good at it.
I'm wondering, like, how to make something like this using the Chimera like surface model, and then you do put that into Illustrator and use color it, get some dots of like lighter shade.
Yeah, I tried to demonstrate a little bit. The first day that I first introduced Chimera, as I was showing some of the lighting controls, I said, I've experimented with using, there's an ambient light setting that can kind of replicate it a little bit, but you really have to tweak the lighting settings, the shininess and lighting settings in Chimera to try to replicate this. I know some people have been able to get pretty close to his style once you get it into a 3D program, which we'll talk about in a couple of weeks. How you take these molecular models and bring them into Maya or Cinema 4D. There's something called a Toon shader that creates almost like a sort of a cartoon 3D rendered model that would look very similar to this. So, I mean, I'm not a big 3D guy, so I don't use that, but I mean, you ask Craig or Glenn, they can show you how to make a Toon shader. I have achieved something kind of close to this in Illustrator. I'll show you an example of that a little later today. But yeah, I do love his style, and so I have tried to emulate it a little bit. Yeah. Well, if you go on the Protein Data Bank, as I mentioned last time, he does the Molecule of the Month feature on the Protein Data Bank. So it's like every time you open up the Protein Data Bank, there's a different molecule, and it's one of his. Yeah. Yeah, these are. He actually has his own molecular visualization program that he wrote himself that I think may kind of give this look. But all of these finished pieces are all watercolors.
So that software that he created, is that just like he himself is the only one that uses it?
As far as I know, I've never asked him whether it's like, hey, Dave, you mind sharing your software because everybody wants to steal your look, your style. That I should ask him that. So here's a different way of illustrating basically that same molecule. This is also the nicotinic acetylcholine receptor. The image at the top was rendered directly in Chimera, or at least most of it was. You can see that it's superimposed on little piece of lipid bilayer that was added in Illustrator. But the ribbon model was rendered directly out of Chimera. And you know, I colorized this one just to distinguish the 5 different chains. Now this one looks actually a little bit different from the one that David illustrated, now that I look at it more closely, because if you look at the top view on the right, the top right image, this is where we're looking down on the molecule from the outside, and you see the arrangement of the alpha helices that are actually embedded in the cell membrane, and the ion pore sits in the middle of that. This one looks like there are 2 alpha chains, but then there's a beta, a gamma, and a delta. So whereas the one that David illustrated, there were 2 alphas, 2 betas, and 1 gamma. So this is a slightly different variant. I mean, what you find is that, you know, first of all, you've got acetylcholine receptors fall into 2 broad categories. There's nicotinic acetylcholine receptors, then there's muscarinic acetylcholine receptors. But even within each of those categories, there are different subvariations that have slightly different configurations of chains, but they all function pretty much the same way. So at the bottom is a really simplified schematic using just simplified shapes created in Illustrator to show the arrangement of those different protein chains, and showing how the binding of acetylcholine, which is that yellow thing that looks sort of like a key in the bottom right. Now the binding of the acetylcholine causes a rotation of an alpha helix, which then opens the pore and allows these sodium ions to flood through. And then that can either initiate muscle contraction if it's a muscle cell, or it can launch an action potential if it's another nerve cell. Yet another example of an illustration showing a ligand-gated ion channel. This is the GABA receptor. GABA is that molecule up at the top shown as a ball and stick. So there's an example of one of my ball and stick models done in Illustrator. GABA is short for gamma aminobutyric acid. It's a small molecule that's a very common neurotransmitter, especially in the central nervous system. And it's an inhibitory neurotransmitter. So whereas acetylcholine is a stimulatory neurotransmitter, acetylcholine opens up channels, allows sodium ions to flood through, which creates an action potential or generates an action potential. GABA does just the opposite. GABA inhibits the generation of an action potential in a postsynaptic neuron. So if you look at this simplified schematic view, you'll see that it also consists of 5 subunits, 2 alphas shown in blue, 2 betas shown in yellow, and a gamma subunit shown in red. There's a different version of that on the right, where there's a delta subunit shown in purple instead of the gamma one, but still basically, 5 subunits arranged in a star shape or radial symmetry. Both of them do basically the same thing. The GABA binds to the receptor on the extracellular side, so outside the cell. The binding of the GABA causes a slight shape change in the molecule, and it opens up a channel. But instead of allowing sodium ions to go flooding through like the acetylcholine receptor, what happens with the GABA receptor is that it allows chloride ions to flow through. That makes the inside of the cell more negative, which makes it resistant to generating an action potential. So it inhibits the firing of this neuron. All right, so those were all ligand-gated ion channels. So you've got a molecule, usually a small molecule, but not always. You've got some kind of ligand, binds to receptor, causes that receptor to do something, or causes it to open up an ion channel and allows ions to flow into the cell. This is an example of another category of receptors known as a voltage-gated ion channel. So if we dissect the name again, it's an ion channel. So somehow this molecule opens up and allows ions to flow into a cell. But instead of being triggered by a ligand, this is triggered by voltage. So a change in the voltage of the cell membrane will then open this molecule and trigger it to then allow ions to flow in, which is going to cause another change in voltage. So this is how an action potential actually propagates or travels down the axon of a nerve cell. So the action potential is generated usually by a ligand-gated ion channel. So you'll have something like acetylcholine bind to a ligand-gated channel, an acetylcholine receptor, which will cause that channel to open up and sodium ions will flood through. That changes the polarity of the cell membrane. So now, instead of the inside being negative, the inside is now more positive. That's what triggers an action potential. But then that change in polarity, it's a voltage. It's a change in voltage of the cell. That change in voltage that was created by the ligand-gated ion channel then triggers these voltage-gated ion channels that are sitting nearby. And then, so they open up, they allow sodium ions to come flooding through. That changes the voltage in that section of the membrane, which then triggers more voltage-gated ion channels nearby for them to open. So they open up and sodium comes rushing in. That changes the voltage in that section of the neuron, and so on, and so on, and so on. That's how the action potential travels down the axon. Is you start the action potential with a ligand-gated ion channel. That changes the voltage, which then triggers these voltage-gated ion channels, which then trigger their neighbors, which trigger their neighbors, which trigger their neighbors. Now, in the case of a myelinated axon, like you see over here on the left, so we've got the neuron cell body, and we've got these green things, which represent pieces of myelin. The myelin acts as an insulator, so there's really no change in voltage happening in the myelinated portions, but all of your ion channels are concentrated in these little gaps called the nodes of Ranvier, little gaps in the myelin sheath. And so the action potential that's generated up here in the cell body, by a ligand-gated ion channel, it causes enough of a change in voltage that it then triggers voltage-gated ion channels farther down in this node of Ranvier. That change in voltage then triggers more voltage-gated ion channels in the next node of Ranvier, and so on. And it creates what's known as a saltatory propagation, where instead of having to stimulate the ion channel, right next door, it's stimulating an ion channel that's like a block away or a couple blocks away, so that the ion channel actually kind of leaps in these little jumps down the axon from one node of Ranvier to the next node of Ranvier, and so on, which dramatically speeds up the propagation of the action potential. So myelinated neurons propagate action potentials more quickly than a non-myelinated neuron. So over here on the right, we have another one of these Goodsell paintings. You see the top of one of these voltage-gated sodium channels. You see the side view over here embedded in the cell membrane. And then on the left is an illustration that I did talking about the channels that are involved in this saltatory propagation of the action potential. And they're really tiny, but if you look in here, there are but some of these little pink guys are voltage-gated sodium channels, which are the ones that then allow the sodium to come rushing in. Right, so these are voltage-gated ion channels that open in response to a change in voltage nearby. Another type of ion channel that's a lot less common, but they're kind of cool, are mechanically gated ion channels. So once again, it's a protein that's going to allow ions to flow into a cell, but it's not stimulated by the binding of a ligand. They're not stimulated by a change in voltage nearby. They're actually stimulated by some kind of mechanical movement of the cell. And a classic example of this is the triggering of the hair cells in the cochlea of the ear. So we've got an illustration of the cochlea on the left, and there's a little cutaway here, and then zoomed way in on the kind of important part of the cochlea. You have these fluid-filled chambers called the scala vestibuli, the scala media, and the scala tympani. And as the tympanic membrane and the ear bones move around, it causes waves of fluid to flow back and forth in these different chambers of fluid. That ends up moving this pink structure here called the tectorial membrane. And that tectorial membrane is in contact with little hairs, little cilia that are protruding out from these cells called hair cells. And on the right, I didn't do this one, but it was the best one I could find. On the right-hand side, you see a real extreme close-up of the tips of those little cilia on the hair cells. And turns out the adjacent cilia, the adjacent hairs on these hair cells, are connected by these proteins called cadherin. And as the hairs kind of sway back and forth in response to the movements of the fluid in the cochlea, in response to these sound waves that are traveling through the fluid in the cochlea, and as these hair cells move back and forth, the cadherin actually pulls on this cluster of proteins down here. This is known as a tip link protein, and it literally pulls the protein open to allow ions to flow through. So I believe it's sodium again that flows into the tips of these little hair cells, and that once again initiates an action potential. And that action potential then travels along branches of the vestibulocochlear nerve to your brain to give you your sense of hearing. So I'm trying to think, and any of my molecular biology experts in here, can anybody else think of another mechanically gated ion channel? This is literally the only one I can think of off the top of my head. All right, so moving away from ion channels, another extremely important group of receptors are the G protein coupled receptors. So these things do not act as ion channels. They don't open up and allow sodium or chloride or something to flow in or out of the cell. These receptors function by a completely different mechanism. Once again, let's kind of dissect the name. Well, it's a receptor, okay, so it's receiving something. It's receiving a ligand, even though the word ligand doesn't appear in the name. The important part here is, though, that they are G protein coupled. That means that the receptor is coupled to or kind of partnered with another group of proteins called G proteins. So here's an example of a G protein coupled receptor. This is an adrenergic receptor, meaning it responds to the binding of adrenaline. Once again, the gray represents the cell membrane, and the pink is the actual adrenergic receptor itself. And there's a little tiny molecule of adrenaline coming here that binds to the adrenergic receptor. But then this whole complex of stuff shown in orange and yellow and light green, this is all a G protein that is bound to the intracellular, the inside portion of that adrenergic receptor. The G protein has 3 subunits. There's an alpha subunit identified here as G alpha. There's a beta subunit shown in yellow, G beta. And then the green one in the back is a gamma subunit, G gamma. So when adrenaline, or whatever the ligand happens to be, you know, there are different ligands for different receptors. When that ligand binds to the receptor, that then causes a change in these G proteins. The G proteins actually break apart. The alpha subunit then will move and bind to some other enzyme, some other protein inside the cell. In this case, I mean, this is, I believe, the most common one. This is an enzyme called adenylyl cyclase, which converts ATP into a phosphate molecule and something called cyclic AMP, or cyclic AMP. So ATP is adenosine triphosphate. It's the nucleotide adenosine that has 3 phosphate groups attached to it. So this adenylyl cyclase breaks off 2 of the phosphates. That's what pyrophosphate is, leaving cyclic AMP, adenosine monophosphate. And that cyclic AMP is an important second messenger that's going to then travel elsewhere inside the cell to then trigger additional events inside the cell. Right. So the key thing here with these G protein coupled receptors is that you've got the actual receptor that's embedded in the cell membrane. The ligand binds to it, and then that causes this complex of G proteins to split apart. The alpha subunit then interacts with some other enzyme, frequently adenylyl cyclase, to then create second messengers. And then you see you've got the beta and gamma subunits are just kind of hanging out after they've been split away from the alpha subunit. Another super important category of receptors in the body are the receptor tyrosine kinases. Kinase is a very common term that you'll see in cell biology, molecular biology. By the way, anything that ends in ASE, hopefully you're all aware of this from your other biology classes. Anything that ends in ASE usually means it's an enzyme. But kinase specifically is a type of enzyme that adds a phosphate group to something else. So these receptor tyrosine kinase molecules are usually these really long, skinny molecules. And if you look at this picture on the right here, you'll see that there's one that's light blue. There's an extracellular component outside the cell. There's a transmembrane component that's embedded in the membrane, and then there's an intracellular component inside the cell. But all of that light blue is all part of one receptor tyrosine kinase molecule. Then the one that's in darker blue is another one. That's another receptor tyrosine kinase molecule, again with extracellular, transmembrane, and intracellular components. Whenever the ligand is present, these receptor tyrosine kinase molecules, they dimerize, meaning they join together in groups of 2. So what you're seeing there is 2 of these receptor tyrosine kinase molecules, the dark blue one and the light blue one, both of them binding to their ligand, which is that purple thing, that or kind of pinkish purple thing up at the top. And that brings them together to form this dimer. And once they've both bound to their ligand and they've dimerized, they join together into a pair of 2. Now the receptor tyrosine kinase molecule has been activated. The name of the molecule comes from the fact that once this receptor has been activated by its ligand, it then adds phosphate groups to tyrosine residues on other proteins inside the cell. Now, remember what the word residue means. We talked about this when I lectured to you about proteins. When you've got a whole bunch of amino acids in a long chain making up a protein, each of those amino acids in that protein chain is now referred to as a residue after it's actually been incorporated into the protein chain. And tyrosine is one of the 20 amino acids that's commonly found in proteins. So what this receptor does is it adds phosphate groups to the tyrosine residues or tyrosine amino acids within other proteins. And what you're looking at here on the left is a picture of the tyrosine amino acid. Now, technically, this is not a residue because it's not actually part of a protein. You can see it's got the whole amino group, the NH2, and it's got its whole carboxylic acid group over here, COOH. So it's not actually incorporated into a protein, but just use your imagination and imagine that that tyrosine was actually part of a polypeptide chain. And this ring over here, this benzene ring, is just the side chain sticking out from that tyrosine residue. So then there's an OH group on the tyrosine that's replaced with a phosphate group. So this is a phosphorylated tyrosine residue in a protein. So that's what these receptor tyrosine kinase molecules do. They phosphorylate tyrosine residues on other proteins. Now, what that does is it essentially activates these other proteins. Whenever you add these phosphate groups to a protein, it activates that protein. That activated protein will then go and probably transfer those phosphates to another protein. It's going to give up its phosphates and phosphorylate something else, which is then going to activate something else. So we'll talk about this in a minute. These second messengers that help carry signals inside the cell, a very common type of second messenger is a kinase protein, which is a protein that just phosphorylates other proteins and kind of propagates this pathway of activation of one protein activating another protein, activating another protein. So you'll see oftentimes in cells, you'll see what are called kinase cascades, where, like I say, one protein phosphorylates another, and then that one phosphorylates a third protein, and so on and so forth. So that's a quick overview of a lot of different kinds of receptors. Like I said, I probably missed some, and I've done a fairly quick job of introducing these receptor types, but hopefully at least you get a sense for the fact that there are different kinds of receptors that are triggered by different things, usually by a ligand, but you can also have receptors that are triggered by voltage, or some that are even triggered mechanically. And then once they're triggered, there are different things that they can do. They can open an ion channel, or they can phosphorylate other proteins, or they can activate G proteins that then trigger second messengers. So speaking of second messengers or secondary messengers, these are molecules that will carry a signal inside a cell, typically from a receptor. It will then carry that signal somewhere else inside the cell. Oftentimes what they do is they amplify the signal. So even if you only stimulate a couple of receptors on the surface of the cell, the second messengers will then actually amplify that signal. So you get more of a larger response from the cell. So this is, a kind of a simplified schematic illustration, but it illustrates some common second messengers that are associated with these receptor tyrosine kinase molecules. Here's I don't know, I'm not real proud of this one, but I'm not crazy about this picture of a receptor tyrosine kinase. It's kind of primitive, but gets the point across, I guess. There's an extracellular domain that binds to a ligand. There's a transmembrane region here just shown as this single alpha helix. And then there's the actual tyrosine kinase domain, which is the intracellular part. And the little loops that look like little handles on that, those are the tyrosine residues sticking out, which they don't really look like that, but it's just, it's supposed to be symbolic. So then in response to the ligand, really this is probably inaccurate because there probably should just be one ligand up here binding to both proteins, creating a dimerization. So you probably wouldn't have two separate ligands. But then the dimerization, the activation, causes phosphorylation of those tyrosine residues. So there's some little red balls here with the letter P inside of them. I don't know how well you can see that on screen, but that represents the addition of phosphate groups to those tyrosine residues. So the receptor tyrosine kinase itself is phosphorylated, but then it's going to transfer those phosphate groups to other molecules. So, for example, it can phosphorylate a protein called phospholipase C, gamma, this little green hexagon looking thing. And phospholipase C gamma is going to actually interact with some lipids in the cell membrane. There's a lipid called phosphatidyl inositol bisphosphate, or PIP2. And the phospholipase C splits that PIP2 into the phosphate head, the polar head of the phospholipid. That's called inositol triphosphate, or IP3. And then the lipid tails are called diacylglycerol. Basically, it's a glycerol molecule with two fatty acids attached to it. And I believe diacylglycerol itself also can act as a second messenger. But in this particular illustration, it's just showing that the IP3 can cause an increase in calcium in the cytoplasm, probably by causing the endoplasmic reticulum to release calcium ions. And then that triggers a bunch of calmodulin-dependent kinases. In other words, proteins that are triggered by calcium that act as kinases that are going to phosphorylate other proteins. And then ultimately what this does is it leads into the nucleus to trigger gene transcription or gene expression. Some other pathways, there's one here that just says other pathways, just because there are others that aren't shown here. There's something called JAK-STAT, where there's phosphorylation of a protein called JAK, which then phosphorylates STAT, which then causes gene transcription, or the RAS-RAF pathway. And I forget what a lot of these things abbreviated in, but Allie or Sheila or one of our experts can probably just rattle off what these things stand for. But so you get RAS, which phosphorylates RAF, which phosphorylates MEK, which phosphorylates MAP kinase. And I think there's like a MAP kinase, if I remember correctly. But again, that ultimately leads to gene expression, transcription of specific genes in the nucleus. So that's what a lot of these second messengers do, is ultimately they just travel to the nucleus to cause the cell to begin transcribing, whatever genes that particular cell type manufactures. One last group of receptors are the nuclear receptors. I mentioned before that many ligands and many molecules cannot cross the cell membrane because the interior of that cell membrane with all those fatty acid tails is nonpolar or hydrophobic. It doesn't like water. But anything that's polar, or anything that has a charge like an ion, can't just pass through the cell membrane. That's why you need pores and channels and receptors and so forth to allow these things to pass through the cell membrane. But there are some molecules that actually can just diffuse across the cell membrane. These would be things that are nonpolar. So oftentimes these are things that themselves are lipids or are based on lipids. Turns out all of the steroid hormones are based on the structure of the cholesterol molecule, which is a lipid. And so all of the steroid hormones, like estrogen, progesterone, and so forth, they are, or cortisol, another one. All of these things are basically lipids. They are nonpolar, so they enter the cell directly by simply diffusing across the cell membrane. Once they enter the cell membrane, or once they enter the cell, once they're in the cytoplasm, then they can bind with a receptor, which then travels into the nucleus and again triggers DNA transcription. So over here on the right, you see an example of the estrogen receptor. Estrogen is shown in light purple down here. It's a lipid that's very similar in structure to cholesterol. That's going to just diffuse easily across the cell membrane. because it's just like the cell membrane, it's also non-polar or hydrophobic. It's going to bind with this, what's called the ligand binding domain of the estrogen receptor in the cytoplasm, and then it's going to translocate into the nucleus. And then inside the nucleus, there's another piece of the estrogen receptor called the DNA binding domain. And the ligand binding domain and the DNA binding domain then interact, and that triggers the transcription of a particular gene or group of genes within the cell. All right, so to summarize what I've talked about so far, we've got passive transport of just having pores and channels that allow molecules to either enter or exit the cell by following their natural concentration gradient, going from high concentration to low concentration, doesn't require any energy. You've got active transport, pumps and synporters and antiporters that pump molecules against their concentration, so pumping them from an area of low concentration to an area of higher concentration. Got all these different kinds of receptor types, ligand-gated ion channels that respond to the binding of a ligand and open up an ion channel, voltage-gated ion channels, mechanical-gated ion channels that respond to different stimuli, but still open an ion channel. You got the G protein coupled receptors that interact with the G protein inside the cell, cause that G protein to split, and it's usually then the alpha subunit that then triggers some other enzyme inside the cell. You've got kinases such as receptor tyrosine kinases, and then you've got these hormonal receptors or nuclear receptors where molecule just enters the cytoplasm directly and then ultimately triggers something inside the nucleus. And then you've got these secondary messengers that transmit the signal within the cell and can amplify the signal, such as cyclic AMP, inositol triphosphate, diacylglycerol, any of these kinase proteins that get phosphorylated, and then ultimately you have transcription factors that activate transcription of DNA inside the nucleus. So one reason that I'm frequently asked to illustrate these kinds of processes is because a lot of drugs function by either activating a receptor or inhibiting a receptor. So a lot of drugs either mimic the action of a natural ligand to stimulate a receptor, or they will block the natural ligand from binding. So I want to talk just a little bit about this because, like I say, this is really important in medical illustration because you may be contacted by a pharmaceutical company to illustrate the mechanism of action of one of their drugs. And some of the examples that I've shown you already in this PowerPoint are things that I created for pharma companies where they wanted to show a particular reaction. And a lot of the other illustrations in here, a lot of these simple schematic ones like this one, these actually come from Netter's Illustrated Pharmacology. So I actually contributed to a Netter book specifically about pharmacology, which pharmacology is the study of drugs. So it was a whole book about drug actions and drug mechanisms. So an agonist is a molecule. It may be a drug, but not always. But an agonist is a molecule that kind of mimics the action of the natural ligand, or what's called the endogenous ligand. The endogenous ligand means the ligand that normally activates a receptor in the body. So on the left, you see that blue thing, which is the endogenous ligand, the normal ligand that activates a receptor. You see it's got kind of a lock and key fit with that purple receptor. When the ligand binds to the receptor, it produces some kind of effect, shown by that glow and the little yellow arrow. But you can have a drug molecule that acts as an agonist, meaning it simulates the action of the endogenous ligand. So when that binds to the receptor, it has the same effect. It activates the receptor. So here's an example of a drug molecule that behaves as an agonist in the body. So what you're seeing on the right is one of these steroid hormone receptors. That's the cortisol receptor. So cortisol is another steroid hormone that looks an awful lot like cholesterol. It's based on the structure of cholesterol. That cortisol can easily, pass through the cell membrane into the cytoplasm, bind with the ligand binding domain of the cortisol receptor. That then translocates into the nucleus, where there's a DNA binding domain, and the two domains interact and trigger gene transcription in a particular gene inside the cell. Dexamethasone is a steroid drug that mimics the behavior of cortisol. So it's an anti-inflammatory drug, so it would be used to help treat somebody with some kind of inflammation, some kind of inflammatory disease or something. So dexamethasone binds to a similar spot on the cortisol receptor and triggers the cortisol receptor. So it mimics the action of cortisol itself. Here's another example. This is the 5-HT receptor, or also known as the serotonin receptor. And again, you see the gray box back there that represents the cell membrane, where this thing is embedded in the cell membrane. And you've got serotonin, which is a neurotransmitter. This would be the endogenous ligand, the natural ligand. Serotonin binds to this particular binding site on the outside of the 5-HT receptor, and triggers it. And this doesn't look like an ion channel, and I believe this is a G protein coupled receptor. So there'd be a G protein inside as well. It's not shown here. And ergotamine is a drug that acts as a serotonin agonist, but is used specifically in the treatment of migraines. So apparently serotonin is a neurotransmitter that's not only involved in things like depression, but it's also involved in migraines. So ergotamine stimulates these serotonin receptors in the treatment of migraine headaches. So these are examples of agonists. The opposite of an agonist is an antagonist. An antagonist blocks the action of the endogenous ligand, blocks the receptor to prevent it from being activated. So again, on the left, you see the endogenous ligand, the normal ligand in the body, binds to its receptor and causes some effect. But if you introduce an antagonist, that antagonist can bind to the receptor, blocking the binding site. The antagonist does not activate the receptor itself, but it prevents the endogenous ligand from binding. So this prevents some kind of action from happening inside the cell by blocking the receptor. So here's an example of that. The picture on the right is an enzyme called cyclooxygenase, abbreviated COX, C-O-X. And there are a couple of different kinds of cyclooxygenase, but I'm not sure which one this is here. But cyclooxygenase is a molecule that's very important in inflammation and the generation of inflammation following an injury. So what happens when there's an injury to the body is if cells are damaged, they may release lipids from their cell membrane. And there's one lipid in particular called arachidonic acid. And arachidonic acid binds to this enzyme called cyclooxygenase. And the enzyme converts arachidonic acid into something called a prostaglandin, which is an important molecule that then triggers an inflammatory response and also causes pain. So there are many drugs known as non-steroidal anti-inflammatory drugs that function by blocking the binding site of cyclooxygenase. So in this case, there's a drug called indomethacin shown in that kind of bright green, that's actually binding to the binding sites of cyclooxygenase, which is then going to prevent arachidonic acid from binding there. So it's going to prevent the conversion of arachidonic acid into prostaglandin. Now, in the cases of these agonists and antagonists that I've shown, so waiting for the screen to catch up with me. I don't know why it has that lag. Okay, so in the case of these agonists and these antagonists, the drug molecule is binding to the actual active site of the protein, of the enzyme or the receptor. So it's either binding to the receptor and stimulating it, in which case it's an agonist, or it's binding to the enzyme or the receptor and blocking it, in which case it's an antagonist. But it's also possible for a molecule to bind somewhere else, not binding to the actual active site, but binding somewhere else on the molecule, what's known as an allosteric site. And binding a drug molecule to an allosteric site can inhibit the rest of the function of the molecule, or it can stimulate the function of a molecule. So here's a real simple schematic showing an enzyme in blue. And you see the endogenous ligand, the ligand that would normally bind to that enzyme or to that receptor and trigger some response. But there's another site somewhere on that enzyme, which is an allosteric site. And you see in the middle panel, what's happened is that you've got some other molecule, could be a drug, that's bound to that allosteric site, and it causes a change in shape to the enzyme or the receptor so that the endogenous ligand doesn't fit anymore. It doesn't bind to it, and therefore it doesn't have its effect. So you're blocking the action of this enzyme, but not by binding directly to the binding site. You're blocking the action of this enzyme or this receptor by binding to an allosteric site. The opposite could also happen. You can also have allosteric activation. Similar kind of thing, but you may have an enzyme or a receptor that is not active. Its binding site is closed, but then introducing some other molecule, like a drug, that binds to the allosteric site might open the binding site on the receptor, and therefore allow the endogenous ligand to bind to that binding site. So you can have something that binds to an allosteric site that turns the protein on, that turns on either an enzyme or a receptor. Okay, so that's a 1 hour overview of cell signaling and cellular interactions, and a little bit of pharmacology thrown in there. We're going to take a break, and then I'm going to show you a bunch of examples in Illustrator of how I've illustrated some of these kinds of receptors and cell membranes and things, but also kind of focusing on how the style has evolved over time. So take a 10-minute break. We'll resume at about 3:20. All right, so as I mentioned earlier, what I want to do is walk you through a little bit of history of illustrating these different kinds of processes, and I'm just going to use lots of examples from my own work and show you how my own style has evolved a lot. But I think that the evolution of my own style kind of reflects the way the whole industry has gone. So even though these are examples from my own work, I think it's pretty representative of where a lot of medical illustrators have gone with this work over a couple of decades. So this illustration is older than almost everyone in this room. This is from about 25 years ago, from a pathology book that I illustrated. And it shows the way most everybody illustrated any kind of molecular or cellular process back then, where all molecules were just represented by simple geometric shapes. And I cringe when I look at this now. I think, oh my gosh, this is so ugly. But it actually it's not all that bad. It works. It communicates the information, which is really the important part. The representation of receptors and other molecular structures has evolved a lot over these past few decades. It's not just to make things prettier, though. I mean, I wouldn't have a problem with this one. It's not the most exciting, the prettiest illustration. It does get the point across, which is the important thing. But so this move away from just using these simple geometric shapes is not just to make the illustrations prettier, but it's also so that over time people will gradually come to recognize some of these important molecules by their shape. So there's been a movement, very gradual movement, but nevertheless, there's been a movement over the past couple decades to create more realistic, accurate looking portrayals of molecular structures, with the hope that eventually we can settle on a bunch of simplified shapes where people can automatically look at something and say, oh, I recognize that shape, that's ATP synthase. Or, oh, I recognize that looks like a receptor tyrosine kinase. Or, oh, I recognize that shape, that looks like a G protein coupled receptor. It's a movement that I heard one medical illustrator refer to as molecular anatomy. The same way somebody could put up a really simple schematic of a liver, and you would immediately recognize that it was a liver just from its shape, or a kidney, or a brain, or a spleen. The goal is to get to that point where people will begin to recognize common molecules based on their shape. Now, you might not be able to tell one receptor tyrosine kinase from another. You might not be able to tell VEGF receptor from HER, human epidermal growth factor receptor, because all tyrosine kinase receptors look very similar, but at least you could tell that it was a receptor that fell into that family based just on its shape. So this is the way things were illustrated a long time ago, using just these simple geometric shapes. Now, just to make this a little bit more of an educational presentation, to get a little bit more use out of this picture, these simple geometric shapes are meant to show an example of a receptor tyrosine kinase. I mean, those proteins don't look anything like this blob that I've shown here in pink. But you look up here, the blue thing is growth factor, so that's the ligand. And you do see here in step 1, after the binding of the ligand, you do get the dimerization, where the two receptor tyrosine kinase receptors actually join together to form a dimer. And you see the P's down here, which represent phosphorylation of those tyrosine residues. And then over here, you see that it begins to interact with some second messengers. So there's phosphorylation of phospholipase C gamma, which is then interacting with that phosphatidyl and inositol diphosphate, or PIP2, in the cell membrane, and splits that into IP3, which is this little ball, and diacylglycerol, which really looks nothing like this. It's just a shape, a geometric shape. That interacts with protein kinase C, and you can see that then ultimately these things lead over to step 8, which is nuclear transcription factor activation, so transcription of genes. But you've got another pathway here. You've got phosphorylation of RAS, which phosphorylates MAP kinase, which then leads to more phosphorylation cascade, which ultimately also leads to transcription factor activation. So showing a lot of the same stuff, just with these very simplified shapes. Here was another attempt. This one's also older than everybody in this room. This was done in 1993. It's one of the first illustrations that I did for a book where we actually illustrated the book entirely in Illustrator. I mean, when I first started out as a medical illustrator, I was still doing a lot of pen and ink work, and even occasional watercolor. But, you know, I had learned Illustrator here from Glenn, and learned a little Photoshop. And so when I got into my first job, I had the opportunity to illustrate a book entirely in Illustrator. In fact, the art director that I was working with at WB Saunders says it was the first book they ever did that was illustrated entirely in Adobe Illustrator on the computer. And so this was an early illustration, just showing in very general way various kinds of proteins and things embedded in the cell membrane. This was for a really elementary pathology textbook. So we get your classic lipid bilayer with our typical blue lollipops. I didn't even have the little zigzags for the lipid tails at this time. They just look like legs on the phospholipids. But we've got, you know, some kind of channel protein here with a channel down the middle, and other proteins. There's carbohydrate chains that have carboxylated the lipids and some of these proteins to create glycolipids and glycoproteins. But still, you know, no attempt to really try to make these things look realistic, basically just simple geometric shapes. And one other thing I'll point out with this one, I think it's even more evident in the next example, is, hey, let's use every color in the visible spectrum just because we can, not because we should. There you go. There's every color in the visible spectrum. This one's actually later. This one's from, I want to say about 2000. So it's still 20 years old, but, you know, maybe a little bit more sophisticated, but still, you know, we got little blue lollipops for cell membrane. At least this time they have squiggly tails, so they look a little bit more like a fatty acid tail on the lollipops. But these various proteins are still pretty much just geometric shapes. I mean, the ion channel has an opening in the middle because it's obviously a channel, but everything else is basically just a simple geometric. I mean, we do have these space filling models up here that I'm a little bit more proud of. I'm a little happier with these. You know, this is done using the exact technique that you used in the previous project of just creating intersecting spheres. But the receptor part of it, still pretty simple geometric shapes. So now we start to get a little bit later. You've seen this one already. I had this one in the PowerPoint. This is this schematic illustration that I did of a nicotinic acetylcholine receptor. I mean, besides the fact that we've got the Chimera models up at the top, the Illustrator model at the bottom, although still a little bit schematic, captures the general shape and also captures the arrangement of the 5 subunits a little bit better. So we're starting to get closer to a little bit more realism in sense of, you know, capturing the 3-dimensional shape and the details of the structure of these molecules. Still got the blue lollipops for lipid bilayer. I'll show you separately the evolution of lipid bilayers and get into that in a little bit more detail, but you'll see some examples of that here. That previous one was for Netter's illustrated pharmacology that I think I mentioned to you. This is one that I did for a pharma company. You saw this one also. So, gosh, I don't even remember now what kind of drug this pharma company was creating, but it was an agonist for GABA. They were creating, you know, GABA is an inhibitory neurotransmitter, and so they were trying to create a drug that mimicked the action of GABA to inhibit something. I can't remember what it was they were trying to inhibit. But again, you know, it's still pretty schematic, but it's actually kind of following the shape of the receptor a little bit. I know for this particular one, I did actually download a file from the protein data bank so that I had something that showed me the approximate structure of the molecule. I simplified it a lot in Illustrator, but still, it's roughly based on the real shape of the molecule. And then, of course, we got the GABA up here. That looks like the project that, not the last project, but the one before that with the ball and stick kind of thing. Notice the lipid bilayer is quite different now. Again, I'll come back and show you more examples of how that's evolved over the years. But getting away from the blue lollipops finally. So this one's probably, I don't know, this is about 10 years old, I think. This one's a little more recent, and you see a pretty dramatic change in style. Sheila or somebody was asking about trying to emulate the Goodsell style. So this is about as close as I've gotten. And this is all done in Illustrator. And so I exported models from Chimera for these, but then redrew them all in Illustrator just using the blob brush tool. So they were pretty time consuming, as you might imagine, trying to render these things with the blob brush, you know, getting all those shadows and so forth. But I think they turned out pretty well. And there is also a lipid bilayer here. There is a membrane, although I didn't bother to show any of the lipid tails because I think they would have been too confusing. But the little purple balls represent the two membranes of the mitochondrion. So you've got an inner membrane and outer membrane in the mitochondrion, just represented by those layers of balls. And what you're looking at here is what's called the electron transport chain, which is a whole bunch of proteins embedded in the inner membrane of the mitochondrion that take things like NADH and FADH2, which are these nucleotide molecules that transport electrons from the citric acid cycle down here to the electron transport chain. So you see you get NADH breaking off into NAD and H+, but with the release of a free electron. And so you've got these electrons that then move from one protein to another. And ultimately what happens here is it's all creating this gradient of hydrogen ions, this proton gradient, because the hydrogen ion is literally just a proton. And then these protons move from this intermembrane space back into the mitochondrial matrix. They pass through this molecule, which is ATP synthase. And that literally drives like a little gear mechanism in here, which then produces ATP. So you get ADP plus phosphorus or phosphate gives you ATP, and that's all being produced by this hydrogen ion or proton gradient. So that's you know, a newer example. Here's another newer one. This is for a pharmaceutical company called Opthea Limited down in Australia. And they were creating a drug for the treatment of wet age-related macular degeneration. And up at the top is a picture of the progression of that disease. So we've got this cut section through the eyeball and a little box in here zooming way in on the macula at the back of the retina. And then up here you see the different layers, cell layers. You've got the nuclear and ganglion cell layers, the photoreceptor layer, which is the rods and cones, and then a layer of cuboidal cells called the retinal pigmented epithelium, and then a thin membrane called Brooks membrane. And then there's a vascular membrane in the choroid, you know, which is just outside of the retina in the wall of the eyeball. And what happens in wet age-related macular degeneration is you've got an overabundance of these proteins called VEGF, which is vascular endothelial growth factor, which is a protein that promotes the formation of new blood vessels. And in the retina, you don't want that. You don't want new blood vessels proliferating because then they cause all kinds of problems. So the blood vessels, they grow, they get leaky, they leak fluid, which leads to the formation of these little pockets of fluid called drusen in the Brooks membrane. And then you got arteries that literally grow up into the photoreceptor layer and begin to distort the photoreceptor layer. So the drug that this company was developing is a VEGF antagonist. It's a molecule that's designed to bind. I forget if it binds to all of these VEGF receptors or if it's selective for just one or two of the VEGF receptors. But if you look at the structure of these VEGF receptors, I mean, if you look down here, you get a little clue to the name or the kind of receptor they are. But just based on the shape alone, I could tell you, and our experts here in class could tell you that these are receptor tyrosine kinases because they have that classic shape, long, skinny molecules with a little skinny transmembrane domain and then this little short intracellular domain, plus the fact that they're forming a dimer, bonded to that VEGF molecule up here. Classic structure of receptor tyrosine kinase. So like I said, once you familiarize yourself with these different proteins, you get to the point where you can start to recognize these things by their shape, which is kind of the goal of this. And by the way, I remember, I don't know, a week ago or so in class, people were asking about pattern brushes in Illustrator. These are all pattern brushes. I think they may have been, it looks like they've been split into components, but I don't know if I still have the brushes here or not. If I do, I'll just demonstrate real quickly. Oh, good. Looks like I do have the brushes. So I've got a brush for the photoreceptors, the rods and cones. And if you just copy and paste these, in front of one another, paste some in the back and paste some more in the back. You can create a bunch of layers of them, and you can create a little effect with a little drop shadow effect. That creates one of these layers, and then we've got oh, and by the way, if you can dissect this brush, you can drag it back onto the desktop, and you can actually see how that's created. This is the artwork here. This is the Illustrator artwork itself to create that repeated brush. So it's 3 rod cells and 1 cone cell. And then we got the RPE, the retinal pigmented epithelium. And that looks like this. It's just 2 little green cells. And then we've got, get the real complicated one. Here's your ganglion layer, nuclear and ganglion layer. And if I drag that out, you see that one's a little more complicated, involves more cells, but it repeats. Once you've drawn it once, then you can get it to repeat, almost forever. And if you modify the path, it just flows with the path. And so that's how I was able to create this distorted these distorted layers of the retina. Just bend the path. And I think there's a couple different variations on that. These are some of the ones that are more in the background line there. So anyway, I'll come back and show you a little bit more about these pattern brushes in a minute. Another example, this one's probably close to 10 years old now too. This is also for Netter's Illustrated Pharmacology, just showing some channel proteins that allow molecules to move passively along their concentration gradient. We've got, I believe this is a voltage gated sodium channel. And then you see these little tiny miniature versions of these different receptors, but this is basically showing the propagation of an action potential. We've got in pink here, we've got a voltage gated sodium channel. So as the voltage comes down the axon, it triggers this to open, allows sodium to flood in, which then continues to propagate that action potential down the cell. But eventually when it reaches the termination of the nerve cell, the action potential causes calcium channels to open. And it's the opening of those calcium channels that then causes the release of neurotransmitters. So then we've got neurotransmitters here in the synaptic cleft. They're going to bind to a ligand-gated sodium channel, which opens that channel and then allows sodium to flow in, which is going to cause depolarization of the membrane of this postsynaptic neuron, which is then going to lead to propagation of an action potential in the postsynaptic neuron. So, a lot of information in there and just, really simplified small versions of these proteins, but looking more like the actual proteins. Every one of these is based off of an actual protein data bank file. Might seem like a lot of effort to make something that's so tiny, but the idea is to create something that's going to be immediately recognizable. Here's a little more complicated version of something similar. This is for Netter's Atlas of Neuroscience. So I did a bunch of illustrations about different neurotransmitters for that book. And this one was specifically about the role of glutamate. Glutamate is an amino acid, but it also functions as a neurotransmitter. So it's released by a presynaptic nerve into the synaptic cleft, where it then binds with a whole bunch of different kinds of receptors. Group 1 glutamate receptors, NMDR, AMPAR, and has different kinds of downstream effects as a result of binding. This TRKB over here is not directly stimulated by glutamate, but it's involved because this group 1 glutamate receptor is actually a G protein coupled receptor. So the G protein stimulates phospholipase C gamma, which is interacting with TRKB. And then that leads to an inositol triphosphate production, which has a bunch of other effects in the cell. So well, the important point here that I want to make is that, once again, every one of these structures is based on an actual protein data bank file. So even though they're fairly simple and schematic, I took the time to make sure that they really look like those molecules. Another one here, this is just another one in that same series from the same book, from the neuroscience book. This one just adds a little bit more detail about some of the second messengers. There's something called calmodulin here, which acts as a kinase, calmodulin kinase, calmodulin kinase 4, MAP kinase, PKA, phosphokinase, all these different phosphorylated proteins that ultimately then behave as transcription factors, or they activate transcription factors inside the nucleus, leading to the transcription of a whole bunch of different genes that are listed over here. Another similar one, I won't go through this one, that looks very similar to one of the others. This is one from one of our former students, Tim Fitzgerald, who lives down in the Atlanta area now. And I believe, if I remember correctly, he did this as part of his thesis work, and created all these little stylized images of different proteins. Well, they're not all proteins. Heparan sulfate's not a protein, but various molecules that are embedded in the cell membrane or that attach to cells. And it, again, kind of emulated that Goodsell style just a little bit. So let me show you some cell membranes. Again, showing how those have kind of evolved over time. Okay. Why are you not changing the font? Oh, that's why, because I clicked the wrong thing. There we go. Yeah, I know. All right, so some of these you've seen already, but I just want you to focus on the cell membrane and ignore the actual receptor molecules. So blue lollipops. So that's just a simple radial gradient. And then the lipid tails just have a simple, also have a linear gradient in them. That's the way I illustrated phospholipids for a long time, but I've been trying to get away from the classic blue lollipop, mainly because, phospholipids don't really look anything like that. More blue lollipops, a little different than the previous ones. I showed an example of this before. At least these have the zigzagging lipid tails. The heads actually consist of 3 different shapes. So when you first open this and you look at it from a distance, it might almost look like there's a little gradient in each of those little lollipops, but there's no gradient in there. I created this using a pattern brush, and you can't put a gradient into a pattern brush. So I did the next best thing, is just used 3 different shapes to create a very subtle illusion of a gradient on each of these little balls, just to give it a little depth. This is a benzodiazepine receptor. So things like Ativan and Xanax and Valium will bind to this receptor. I mean, that's not really what they're supposed to do in the body. I forget what kind of receptor it normally is, but it's activated by these benzodiazepine drugs. And you see, it's very similar to some of the others I've shown you, where it's 2 alpha, 2 beta, and a gamma subunit arranged in a kind of a 5-sided star shape. You've seen this one before, but now focus on the lipid bilayer. No blue lollipops. Was trying to go with something that looked a little bit more like an actual phospholipid. So it's, the phosphate group is actually quite small in a phospholipid. It's not the great big blue lollipop head that you always see. So the yellow here is meant to represent a phosphorus atom, and then the pinks are oxygens, and then all these gray circles represent carbons in this branch, or in this zigzagging carbon chain that makes up the fatty acid tails. So I felt this was a little more realistic, but in retrospect, looking at it, the only thing I'm not crazy about is it's a little too busy, a little too much going on in that lipid bilayer for my taste. So it's my lipid bilayers have continued to evolve beyond this. And now this is closer to what I do now, where I've got something that's a little bit more monochrome, and where the lipids in those fatty acid tails, rather than having just a bunch of separate balls where you see all the outlines between them. So instead of you know, instead of this, where you have all these overlapping balls to create the zigzagging chain, they've all been fused together into one. And whoops. That's not what I wanted to do. So, you know, if you have a bunch of circles like this, and you want to merge them all into one shape, you need to bring up your Pathfinder palette, select them all, and choose this unite option from the Pathfinders, and it merges them all into one shape. So that's how I created these lipid tails. It's just a bunch of circles, and then use the Pathfinder. So I feel like that's still capturing a little bit more accurate sense for what a phospholipid really looks like, but without being as busy as the one that I showed you previously. So over the years, as I've experimented with different kinds of patterns or different kinds of brushes for phospholipids and lipid bilayers, I've kept a file on my computer that I still have on my computer at home, where I've maintained a whole collection of these different examples, as you know, that I've saved them all as pattern brushes. So this is a pattern brush over here on the left, and you can confirm that by just, you know, moving it around. You see that all the little shapes reflow. This is the basic structure of that, and it's just, you know, a blue lollipop with a couple other shapes just to give it a little sense of form, you know, a little subtle highlight. And then our phospholipid tails, or excuse me, lipid bilayer tails. And actually, I drew this once, and then just used the reflect tool, which is hiding under here, reflect it across a horizontal axis and click copy. And then, now this part, the only thing I don't like about this is that the highlight is now facing down. But if I just reflect it again across a horizontal axis, now the highlight's facing to the upper left. And I could take these lipid tails and reflect them across a vertical axis. So then all I got to do is, what do I do with my brushes? I think I closed my brushes palette. It's my brushes palette. I just select all these shapes, drag it into the brushes palette, and say make a new pattern brush, and give it a name. I probably got about 4 of these in here, so we're going to call this newest lipid bilayer, something like that. So once you've done that, you can then draw any path. Get rid of the fill color. Go to the stroke, select that path, and you get something that automatically flows to create the lipid bilayer. Now, what you'll find is if you create a really tight corner, you know, things get really distorted. So you don't want to do that. But as long as you give it a fairly gradual curvature, when you're zoomed out, you don't really notice the distortion very much. Especially if I flatten this out even a little bit more, you can hardly notice any distortion. But then these things have evolved over time. You know, here's an early attempt where we've got, you know, all these little gray balls plus an attempt to do the phosphate ends up there. Just drag it into your brushes palette, make it a pattern brush, call it lots of gray balls, something like that. So then you can select that path. Let's now apply that path to it. Same thing, you got to be careful. You can't distort it too much, because otherwise you get really weird, you know, distortion of the objects. And then the newer ones are down here. You could just take one of these and turn it into a pattern brush. Just one. But then you'll see what happens when I apply that to my path. I mean, it might look okay, but it's very, very stiff and repetitive, I feel like. So what I'll do is I'll create a little variety. Have some that bend one way, some that bend another way, just to create a little bit of variety in the phospholipids. Because that's realistic, you know, that you've got different fatty acid tails, so there is actually some variation. So what I'll do is I'll grab this whole group, drag it in here, and so now that whole thing becomes a brush. And so that whole group of, you know, however many was, 10 or 12 phospholipids repeat. So you get a little bit more variation in the tails. It just looks a little bit more natural, a little bit less stiff and mechanical. And last thing I wanted to show you is that something that I kind of enjoy doing in some of my figures is to create a lipid bilayer where we don't just see it cut on the edge, but we also see kind of the top of the lipid bilayer as if we're looking at, you know, the outside of the cell, but it's beginning to fade into the distance a little bit. And if you have a background color behind your cell, like for example, you've probably heard me say before that I don't normally like background colors unless they serve a purpose. But we might have a light blue background back there, which would represent the extracellular fluid. And then I might actually have a different color down here, like make this, I don't know, maybe make this a little green to represent the cytoplasm. So in this case, you know, having a little blue outside the cell and a little green inside the cell actually does serve a purpose because we're distinguishing the extracellular fluid from the cytoplasm. But the point here is that to make that cell membrane look like it's fading into the distance, that tan color that we see in the foreground has to gradually fade over a series of steps to that light blue color in the background. And one way you can do that is I've actually got I've got this, let me see. Oops. There's our cut edge of our lipid bilayer. So we're seeing both the outer layer, you know, up here. You know, so this would be the outer layer, and then this would be the inner layer. But you know, you can put those together to create one pattern brush to get both the outer and inner layers of the brush. So that's what this object is right here. This has got, this has got, you know, both inner and outer layers. The part that's fading into the background, though, you really only need the outer layer, you know, because we're only looking at the outside of the outer layer, you know, disappearing into the background. So what I can do is Can I actually get rid of that? Make this a little smaller. Oops. So I can create a new brush, which is just the basically just the phosphate head, just the head of our phospholipids. I don't like the fact that the ones in the foreground, there's actually space between the heads, whereas the one behind it, they're touching one another. That's because I don't have any tails on the one in the background, but I can add space between those. If I double click on the brush, I can add spacing. And the percentage here, it's a percentage of the width of the object that makes up the brush. So the object that makes up this brush is that tan ball. So if I put 50% in here, it's going to put space equal to half the width of the ball. I probably don't want that much. Let's try maybe 20%. So you can see what it's done is it's added a little space. Then what I can do is I could copy and paste and back. Just nudge it over and nudge it up a little bit. Same thing, copy, paste and back. Nudge it up, nudge it over. If you keep doing that, copy, paste and back, nudge it up, nudge it over. You can start to create this sense that the cell membrane is disappearing into the future. Now it's not fading yet. I haven't changed the color, but at least I'm creating the sense that the little balls are extending away from us. So by the way, if you're not familiar with paste and back, I'm doing copy and then paste and back or Command B. I'm just using the arrow keys on the keyboard to nudge that up, nudge it over a little bit. And same thing, copy, paste and back, up and over, copy, paste and back, up and over. And you can do that in like 30 seconds. You can get a whole bunch of these layers of little balls going off into the distance. But if you want to create a faded color to get that to look like it's fading into that blue background, we actually need to create a whole bunch of separate brushes with different colors in them. And the way I did that is I actually started out with an object that's colored my tan color. And then I tried to imagine, all right, what would it look like if that tan ball were almost completely faded into that blue background? So if I look at my CMYK breakdown here, here's my tan ball. It's just 15 magenta, 40 yellow. If I look at my blue background, it's 10 cyan and 5 magenta. So this has got to be pretty close to 10 cyan and 5 magenta, but maybe with just a little bit of yellow added. So there it is, just 10 cyan and 5 magenta. But if I just throw a little bit of yellow in there, it's like a slightly yellower version of that background color. And then here's a little trick. I'm going to create an object blend between them. I'm going to select both of those shapes, say object blend make. Now what it's done is it's created something that looks like a continuous transition between those two shapes. But if I go back up here and say object blend options, I can say instead of giving me a smooth color, I can say give me a specified number of steps, And I could say, all right, let's make 8 specified steps. Click OK. Now I can once again go back up to Object, Blend, and say Expand. And each of these is now a separate shape. And they are intermediate in color between the foreground object and the background object. So that using an object blend like that, and then expanding into component shapes, is a great way to create a series of shapes that transition gradually from one color to another. So what I could do now is I could just take each of these objects and drag them into my brushes palette and call this one like ball 2. Take the next one, drag it in there, pattern brush, ball 3, ball 4, and so on. And I won't do them all. But so what that means is I've got a bunch of little balls that are gradually changing in color. So if I came up here, for example, I could color this one with ball 2. The size is different, so it's not looking exactly right. Color this one ball 3. Next one, come on. Next one ball 4, and so on, and continue until I got all the way to whatever it is, ball 8 or 10 or whatever. And by that time, it would look like it was just fading into that blue background. So it would look like this, and then if you put put that blue background behind there, kind of looks like our membrane is fading off into the distance. Okay, so that's evolution of representation of receptors and so forth, as well as representations of cell membranes. Let me talk a little bit about the next assignment that's going to utilize both of these things. So I'm asking you to create an illustration showing a receptor interaction, and I want you to show if it's a ligand gated channel or something like that. I want you to illustrate the ligand, show the ligand binding to the receptor in the appropriate place. I can't remember. At least one of these is a G protein coupled receptor, so it wouldn't actually behave as a channel, but you'd want to show the G protein inside, and then I want you to actually illustrate a little bit more, just to create an illustration that kind of talks about what's going to happen as a result of the binding of the ligand to this particular receptor. How is the cell going to respond? What kind of second messengers might be involved? Is it going to promote gene transcription, or is it going to cause some other thing to happen inside the cell? So I've given you a list to choose from here: the nicotinic acetylcholine receptor, a muscarinic acetylcholine receptor, which is a little different, and then a handful of others. I've actually given you the PDB ID number for each of these if you want to use it. I'm open to suggestions if people have other ideas of a different kind of receptor that they want to illustrate. I did put a little note in here that if you want to try something challenging, you can try to illustrate a receptor tyrosine kinase, like human epidermal growth factor or insulin receptor. The problem with these is that you're not going to find just one protein data bank file for them, because to my knowledge, nobody has ever solved the entire structure of a receptor tyrosine kinase all in one model. Now they've solved the extracellular domain, they've solved the transmembrane domain, and they've solved the intercellular domain. But that would involve retrieving three different protein data bank files and then figuring out how they relate to one another. But feel free to do that if you want that extra little bit of a challenge. Once you've picked a molecule to illustrate, a receptor or a receptor process to illustrate, I want you to do a little research on it in the protein data bank. So if I were to choose, this number like this 5KXI, if I go to my protein data bank, and you can use the link that I have in my courses, or eventually you just get so you memorize it, rcsb.org. And then if we enter that PDB ID number in here, we say X-ray structure of human alpha 4 beta 2 nicotinic receptor, and it's a nicotinic acetylcholine receptor. I want you to read about it a little bit. I want you to understand what it does. I want you to understand a little bit about the protein data bank entry. There's actually information here about the article in which this structure was first discussed. So you could actually go and try to retrieve that Nature article, which you should be able to do from RIT's online library. But there's also an abstract right here, so you can open the abstract and you can read just a synopsis of what that molecule does and how it behaves, how it functions. Also, check out information about the molecule down here, about the PDB entry. Now this is only subunit alpha 4, it's only chains A and D. It's two of the chains of the molecule in entity ID number 1. But if we scroll down here, we see that, oh, here are the other 3 chains, the beta chains, the B, C, and E. So we've actually got chains A, B, C, D, and E. We've got all 5 chains of this particular receptor, because it's one of these that consists of 5 subunits. So all of that is included in here, but then keep scrolling down to see if there's any other information in here. There are some ligands, although these are not the endogenous ligands. You'd have to, research and figure out what the heck these things are, but none of these are acetylcholine. So none of these are the normal endogenous ligand for the nicotinic acetylcholine receptor. I mean, you'd have to kind of research what these things are to know that, okay, that's not nicotine. You could go to one of the other databases like PubChem, and you can look up, oh no, nicotinic, not, it's a nicotinic acetylcholine receptor, but we want acetylcholine. By the way, the reason that the nicotinic acetylcholine receptor is called nicotinic is because not only does it respond to acetylcholine, it also responds to nicotine. And the other type of acetylcholine receptor is called a muscarinic acetylcholine receptor, which interacts with a molecule called muscarine, which is a drug that's found in certain mushrooms. But here in PubChem, we can get our model of acetylcholine. Remember, don't download the 2D structure. You want to come down here to the 3D conformer, download that, save it as an SDF file. And if we open that in Chimera, see, that's what acetylcholine looks like. So as part of the assignment, you have to illustrate this as well. You can illustrate it as a ball and stick, or you can illustrate it as a space filling model, but use Chimera or PyMol to generate a reference, but you're going to want to actually illustrate it in Adobe Illustrator. The whole final illustration is going to be done in Illustrator. So going back to the protein data bank, we can find out that, all right, there's some other molecules in here, but none of these are actually acetylcholine. So if you render this molecule in Chimera and then bring it into Illustrator to draw it, you're going to want to probably get rid of those other molecules because they don't really have anything to do with the normal functioning of the molecule. One last thing here in the, oh no, never mind. I was going to say, remember to download the biological assembly. Don't just download the basic PDB file, because that may not give you the whole molecule. You want to download the biological assembly. But then I remembered, you're actually not going to download it from the protein data bank at all. The protein data bank is there just for you to do research on it. If you're going to illustrate a receptor that's actually embedded in the cell membrane, you really want to download it not from the protein data bank, but from the OPM database. So that PDB file was 5KX1, or maybe that's an I, I'm not sure. We go to the OPM database and enter that same PDB number. Sure enough, we get that molecule pops up in the OPM database. I'm going to download the OPM file. And open that in Chimera. Okay, so here's our nicotinic acetylcholine receptor opened up in Chimera. But the reason I want you to download this from the OPM database, the Orientation of Proteins and Membranes database, is because remember, if we select structure protein, I'm going to hide atoms and bonds, but then I'm going to select and invert to select everything else. Now it's selected some of those ligands that we don't want. I'm just going to leave them in there for now. But I'm going to say atoms and bonds show, and we get the rows of red dots and blue dots, which tells us how this thing sits in the membrane. Remember, the red dots represent the outside of the cell membrane, and the blue dots represent the inside of the cell membrane. So that's going to tell us where this thing sits in the membrane, so that we can actually illustrate this thing sitting in the correct place in the cell membrane. So if I go back here to the actual assignment, so use the PDB ID number to kind of research the molecule in the protein data bank, but I put it in bold and italic, so hopefully nobody will miss it. Download it from the OPM database, because that way you'll get those layers of balls so that you know exactly where the thing sits in the membrane. So then you'll want to generate some references in Chimera. I'm going to try to, let me go back and select the protein again. Add a surface to it. Cross your fingers that it works. Yeah. Oh, it did work at least. Did it get all of it? Yeah. And I don't know why it keeps giving me error messages, even when it works. I am going to change one thing. You might be able to tell that we're looking at this in perspective view. Probably better off in our viewing controls under camera. Probably better off switching this to orthographic. That way we're looking straight at it. And as I rotate it, a little bit to get those layers of red and blue balls perfectly straight. You know, I see this is what the thing looks like. We're looking at it directly from the side. I probably want to go ahead and save an image. And it doesn't really matter, you know, like what format, but you don't even really have to use the print units, but I tend to do that. I'm just going to go ahead and save. I don't need this to be a really big one. So, you know, 8 by 4 inches, that's fine. Doesn't have to be 300 pixels per inch because this is not going to be our final output. This is just going to be for, you know, for a reference that we're going to redraw in Illustrator. So I can save that. I could try to play with the lighting and maybe get a little bit more, I don't know, maybe a little more dramatic lighting on it or something. Just so that when I render it in Illustrator, I get something with a little bit more shading and form on it. So the JPEG that I just exported is, you know, shows the overall outside of the molecule and shows where it sits in the lipid bilayer. But a lot of these molecules, a lot of these receptors are ion channels. They are, you know, like ligand-gated ion channels, or maybe a voltage-gated ion channel. But that means they're going to have a channel in the middle that runs through the middle of the molecule, and that's where our ions are going to pass through. So you might also want to illustrate some of these channels in cross-section. And there's an interesting thing you can do in Chimera that I haven't shown you before, but under these viewing controls, remember that's under Tools, Viewing Controls, there's a thing called Side View. And what this is showing is the little yellow square on the far left represents the viewer's point of view. And then these lines, these other yellow lines, represent planes that we can use to crop into the molecule. So if I take this left-hand yellow plane and drag it into the molecule, I can actually cut into it and generate a cut sectional view that shows the pore that runs down the middle of the molecule. And so I could export another image, you know, call it cutaway or something, exact same size. And that'll should be on my desktop. And so I've got a cutaway view so that if I wanted to illustrate a cross section of this molecule so that we could actually see that little ion pore, ion channel in the middle, we could do that. So you'll export some images from Chimera, and you notice that I kept the little red balls, the little blue balls when I exported those, so that, you know, when I redraw this in Illustrator, I can you know exactly where the receptor sits relative to the cell membrane. So let's see here. You know, a lot of this just kind of walks through what I was just talking about. Here's the Tools, Viewing Control, Side View to create a cross-sectional view if you're illustrating an ion channel. And then you're going to illustrate the receptor and the process in Illustrator. So if I were to go back into Illustrator, I'm going to create a new file. And just to be safe, I always pick from the print presets, assuming that I'm creating this for print. And if you look at the specifications, this is going to be done in CMYK color, so it's intended for print. So if I choose one of the print presets, choose letter size. Over here on the right, you see it's going to open this as a CMYK color document. This is important because, I had a couple people in the previous project who did theirs in RGB when it was supposed to be CMYK. And, make sure the raster effects is set to high. You may not use any of those effects. You might not use drop shadow or inner glow, but if you do, you want to make sure that this effect setting is set to high. And so here's our document. So then I'm going to use the place command to place one of those images in here. And so I could use that just as a template. I'm going to, in my layers palette, I'm going to lock that, create a new layer. And I can use this to illustrate my molecule. And there's a couple of different approaches you can take to illustrating this thing, but I tend to like the blob brush. And you can control, notice with the blob brush tool, you can see a circle, which represents the diameter of the brush. Let's see, I'm going to give this a bright color so that I can actually see it when I'm painting. But the circle represents the diameter. If I wanted to get something that was closer to the size of these little balls that you see here, I can reduce the size of that circle just using the bracket keys on the keyboard. So left bracket makes it smaller, right bracket makes it bigger. So I can just come in here and just start painting with the blob brush, and I don't even have to be super careful. It doesn't have to be super accurate because this thing's only going to be like 1/2 inch in size in the finished product. I could probably even paint this with a bigger blob brush because you're just not going to see this level of detail at this scale. But kind of committed now. So even though I picked the brush up a couple times and moved around, it's still going to create one continuous shape. And if I select that whole thing now, I can use the white arrow and actually delete the interior shape. And that leaves me with just the exterior shape. And then the tricky part then becomes trying to paint like some of the shadows and highlights. I mean, something I could do is I could temporarily hide this layer, add yet another layer, and then come in maybe with a darker color. I'll probably change these colors later because I'm not crazy about these colors, but I could come in here with a little darker color and start painting in, some rough shadows to capture, where some of these shadow shapes are located. I'm trying to emulate that Goodsell look a little bit, like I did with the that illustration I showed you that I did of the electron transport chain. Trying to emulate that a little bit. You don't necessarily have to work in the Goodsell style, but I do kind of like that look. So anyway, just painting in some shadow areas. And I'm doing this, you can tell I'm doing it very, very roughly, very approximate. Then I turn that other layer on and I start to see what some of that looks like. And I could add even, maybe a third layer. And now go with a lighter color and start putting in, some highlight areas, although I might want to again use my illustration as a guide for where some of these more prominent highlight areas are going to be. And I'm obviously not doing this with a great deal of care. I'm being kind of sloppy now. I'd be a little bit more precise if I wasn't rushed for time. But you start to get that, and I'd tweak these colors a little bit. So you can create, an external view of the molecule. Like I say, you'd want to devote a lot more time to it and be more careful. But then I'm going to hide my background where that artwork was, and I'm going to put in my lipid bilayer. And I guess it would probably help if you're going to have to create your own lipid bilayer from scratch, but I have the advantage of having a bunch pre-created. My lipid bilayer brushes, probably pick this one. So I can just copy the shapes that are used for the brush, paste that in here, and then drag it into my brushes palette. Give it a name. And let me just turn my reference image on again. I can see where the red and blue balls are located. So I know my cell membrane is going to be kind of in here somewhere. And you apply that lipid bilayer to that path. And that's relatively close. I just kind of got lucky that it happened to be just the right width. So now I'm going to drag that shape to the background and send it to the back. And I might want to make my lipid bilayer look like it's kind of fading into the background. Something that I'll frequently do is I'll draw another path. And rather than giving it, a pattern brush, I'll just give it a color and then make it a much thicker stroke. And send that to the background. Oops. Oh, helps if you put it on the right layer. So then that becomes kind of like almost like a shadow in the cutaway of the lipid bilayer. And then I could take these balls and let's see, maybe I will try to make it look like it's fading. So I don't have a background. I haven't put a background color in here yet, but I'm just going to imagine that I'm going to use that same blue. So we'll do, say, 15, 5, and 10. Whoops, and 10. Might do the same thing with the stroke on it. I might make that stroke, give it a little bit of cyan and reduce the amount of magenta, yellow, and black, so it's not such a dark stroke. Let's see. So now I've got something that has a fill, which is almost the same blue as the background that I'm going to use. And then I've got this tan one with a darker outline. So I'm going to object, blend, where is it? Make, object, blend, options. Specified steps, let's try 8 again. And then one last thing, object, blend, expand, splits it into its component shapes. And I can then drag each of these individually into my brushes palette. So this is going to be all one. Ball 2. Oops. Ball 3. And once again, I'm not going to do them all, but oops. Five. So then I'd want to take this basically the same path, copy it, paste in back, which is Command B. Now there it is back there, but now I'm going to put this ball 1 path in there. And once again, I don't like the fact that the phosphate heads on the lipid bilayer in the foreground have a gap between them, whereas the ones in that other path are touching. So I have to just double click on it, put some space between them, like 20%, something like that. And I'm going to have to do that with all of these. So then I can select this one, copy, paste in back, move it up, nudge it over a little bit, but give it ball 2 color, copy, paste in back, nudge it up, move it over, ball 3, copy, paste in back, move it up, move it over, ball 4, copy, paste in back, move it up, move it over, ball 5, and I continue to do that, until I got through all 10 of these to make it look like it was fading into the background. I might need to make that dark brown line a little thicker though, to hide that gap. So I'm making it a little thicker, and I'm also going to nudge it up a little bit and then send it all the way to the back. I don't know, might not want it to be quite that dark or brown, but anyway. So that's giving you the rough idea. Now you can go back here. That could be this could be one version of the receptor. You might illustrate the outside of it. I see. Need another layer. So I might, have this over here. Oh, but make sure it's right. I might, since I've got a little curve to my lipid bilayer, I might want to rotate that very slightly to follow the curvature. And then go back on my layer 1 here that has my reference artwork and place my other image with the cutaway and do pretty much the same thing, but have a a flat surface have a flat surface for this cut plane. And not going to draw the whole thing. These molecules are what did I just do? Huh. It's weird. I swear I drew this thing and then it vanished on me. Mysteriously. Anyway, I draw the whole thing. Okay. Why the hell does it keep doing that? All right, it's there. Must have just been hiding behind something else. One thing you can do with this cutaway. Why is that not showing up? All right, something really weird's going on with Illustrator here. So this, oh, I see what I did. All right, I'm not smart. There we go. All right, so, down here, I could, finish drawing one half of this thing. Again, I'm not going to be real careful about it. But these things are more or less symmetrical. So if you wanted, you could just use the reflect tool and just copy to make a mirror image of it. And then you'd probably draw like a darker area in the middle to represent the pore of the channel. And then all right. Illustrator is really doing some weird stuff here. I don't think it's user error, but it could be. Okay. Very strange. Right. I know we're going to have to go soon because we're running out of time, but so just use your imagination and imagine that I had drawn another version of this receptor over here that was cut away. And of course, I'd have to, tell the story. I'd have to show what actually is happening here with the binding of the ligand and so forth. But one final important thing, I've gone back and forth with this particular assignment a couple times about the size of the assignment, the size of the final piece. And if you read the specifications carefully, it's really small. The final illustration is only going to be 20 picas in width. And I hope everybody in here knows what a pica is from their computer applications class. I'm going to go into document setup, and the units are set to points, or I can change it from points to picas, but the entire width of this illustration, the final thing, is only going to be 20 picas in width. So I don't actually create my artboard at that size. There's no reason to create the actual document at 20 picas in size. I still work on a standard 8.5 by 11 sheet of paper here, but at the very end, I'm going to create a rectangle that's 20 picas in width. And I also say, I don't care how tall it is, but I did give you a maximum height, maximum of 40 picas in height. So that's 20 by 40 right there. So if I make this my topmost object, well, let's see, let's do this. I'm gonna drag it up to the top. I'm gonna give it no fill. I'm gonna give it a black. Come on. What are you doing? There we go. Give it a black stroke. So I need to fit the artwork in that little tiny space. I'm gonna lock that object temporarily. And then I'm going to try to resize this artwork so it fits in that little space. Now I can scale, whoops, shit. Pardon my French. I got something locked that I forgot to unlock. All right, let's try that again. Lock that, select everything, scale it. So I can scale this down to fit in that 20 pica space. The problem is, you might notice the strokes aren't scaling. There is a setting that you can choose to make sure that they do scale. When I use my scale tool, there's a checkbox here, scale strokes and effects. So I'm going to turn that on, and that way my pattern brushes will actually get skinnier and they'll scale as I scale everything else down. So now it fits within my 20 pica width. The little balls, the lipid bilayer scaled proportionally. You know, I might add a blue background. Back there behind, I'm going to put it behind the, so membrane. I might have a different color for the cytoplasm, you know, like I showed before. Maybe use a green for the cytoplasm. And then very last step would be to select all of this and make it a clipping mask. Object, clipping mask, make. And now that has ensured that it cropped it to the exact width that I wanted of 20 picas. So again, I'm still working on an 8.5 by 11 document, but I've cropped it to the exact width of 20 picas, which is in the specifications. So there's also a requirement to add labels, because you're going to be describing a process. So you want to add some text labels. You know, look at the assignment sheet, the specifications for the text that's in here. This might seem very tiny, this illustration, but This is the width of a standard column in a textbook. And, you know, you should be able to fit this information into that 20 pica width. Okay, sorry, I ran over a little bit, but so try to get something done on this before Monday so that you actually have an opportunity to get some feedback from me during Monday's class.
Jim.
Yeah.
Like color, do you usually go with, whatever color the protein or model or whatever that comes up in, like, you can do it in Chimera or anything, or do they, or is there like certain colors that are associated with, you know, a given